Have you ever wondered how a technology developed inside a laboratory reaches farmers, industries, and the market without losing the inventor's rights? Or why researchers sign agreements before sharing seeds, microorganisms, or new inventions? In this session, we'll understand licensing of technologies, material transfer agreements, research collaboration agreements, and license agreements in a simple and exam-oriented way. By the end of this lecture, you'll clearly understand how intellectual property is legally protected while promoting innovation. The first objective is understand technology licensing fundamentals. This will help us understand what technology licensing is, why it is needed, and how it works. The next objective is learn about material transfer agreements, or MTAs. We will understand when an MTA is required and its major components. The next objective is study research collaboration agreements, or RCAs. These agreements help different organizations work together on research while clearly defining their responsibilities. The next objective is know the specifics of license agreements. We will also understand the important elements involved in a license agreement. Finally, explore practical agricultural applications. This will help us relate these concepts to agriculture and real-world technology transfer. Now, let us understand the introduction to IP commercialization. The first point is commercialization, moving laboratory innovations into the societal domain for public benefit. In simple words, technologies developed through research should reach society so that people can benefit from them. The next point is legal protection, safeguarding the intellectual property rights of inventors and institutions. This ensures that the ownership and rights of the innovation are legally protected. The next point is technology sharing, fostering collaborative growth through legal and structured exchange models. This allows technology to be shared safely between organizations through proper legal agreements. The final point is agricultural growth, driving modern farming practices through high-tech research and development. This helps improve agricultural productivity by bringing advanced technologies into practical use. Now, let us understand what technology licensing means. A contractual arrangement where the intellectual property owner grants permission to another party to use their technology for commercial exploitation. In simple words, the owner allows another organization or company to use the technology under specific legal terms. Now, let us understand the two important parties involved. The first is the licensor, the legal owner of the technology, usually universities or ICAR institutes, grants rights in exchange for royalties. This means the licensor owns the technology and permits others to use it while receiving royalty payments. The second is the licensee, the party acquiring the usage rights, usually private companies or startups, responsible for scaling and marketing. The licensee takes the technology and converts it into products or services that can reach the market. Now, let us understand the need for technology licensing. The first point is commercialization. Ensures that laboratory innovations reach the market and benefit the public. Research becomes truly valuable only when it reaches society. The next point is wider adoption. Facilitates the large-scale implementation of agricultural technologies. Technology licensing allows useful innovations to be adopted by more farmers and industries. The next point is revenue generation. Provides a steady and sustainable income stream through royalty payments. This benefits research organizations financially. The next point is industry partnership. Bridges the gap between academic research and industrial application. It creates collaboration between researchers and industries for practical implementation. The final point is innovation promotion. Encourages continuous improvement and new discoveries in agriculture. Technology licensing motivates researchers to develop more useful innovations. Now, let us discuss the types of technology licensing. The first type is exclusive. Only one licensee is granted the right to use the technology. Here, no other organization receives the same licensing rights. The next type is non-exclusive. Multiple licensees can use the technology simultaneously. This allows the technology to reach a wider market. The next type is sole. Only the owner and one licensee can use it together. Both parties can use the technology at the same time. The next type is cross-licensing, mutual exchange of IP rights between two parties. Both organizations allow each other to use their IP. The final type is compulsory, government-mandated licensing for public interest or health. This type is mainly used when public welfare becomes more important than exclusive ownership. Now, let us understand the technology licensing process. The process begins with innovation, discovery, and development. Every technology starts with research and innovation. The next step is IP protection, filing patents or intellectual property rights. Legal protection is obtained before sharing the technology. The next step is evaluation, market assessment. The commercial potential of the technology is evaluated. The next step is negotiation, finalizing terms. Both parties discuss and agree on the licensing conditions. The next step is agreement, legal contract. The agreed terms are documented in a legally binding agreement. Finally, commercial, market launch. The licensed technology is introduced into the market for practical use. Remember this sequence because it is commonly asked in examinations. Now, let us discuss the key advantages of licensing. The first advantage is faster commercialization. Significantly speeds up the process of bringing laboratory innovations to the market. Technology reaches industries and society much faster. The next advantage is increased revenue. Provides a steady and sustainable income stream through royalty payments. This benefits research organizations financially. The next point is reduced investment risk. Shares the financial burden and risks associated with product development and scaling. Both parties share the responsibility instead of one organization bearing the entire risk. The final point is market expansion. Enables technology to reach new geographic regions and diverse market segments. This increases the overall impact and adoption of the technology.
Moving further, let's discuss the common challenges in licensing. The first challenge is valuation issues. Hard to price early-stage innovations accurately. The next challenge is legal disputes. Conflicts from unclear contract terms. Another challenge is confidentiality risks. Protecting trade secrets during disclosures. Following that, royalty negotiation. Agreeing fair and sustainable payments. And finally, technology misuse. Ensuring licensee adheres to permitted use. Think about this for a moment. Every advantage we just discussed comes with a corresponding challenge, and that balance is exactly why licensing agreements need to be drafted so carefully. Let's continue with our next major topic, Material Transfer Agreement or MTA. What is an MTA? A legal contract that governs the transfer of tangible research materials between two organizations. This covers biological samples, chemicals, and software, protects the IP of the provider, defines the permitted scope of research use, ensures legal clarity for both parties. So essentially, while a license agreement deals with the rights to use technology, an MTA deals specifically with the physical transfer of research materials. One more important concept is, when is an MTA required? The first situation is seed exchange and crop varieties. Next is microorganisms and cultures. Following that, cell cultures and biological lines. Another situation is plant materials and germplasm. And finally, genetic resources, that is DNA or RNA. Can you relate this to a real-life example? Think about a university sending a new pest-resistant seed variety to another lab for testing. That transfer would require an MTA. Let's move to major components of an MTA. If you observe the illustration, there are 6 major components. The first is provider details, full legal name and address of the source institution. The next is recipient details, information on the user and the receiving laboratory. Another component is material description, specific details of the biological or chemical material. Following that, usage conditions, defined scope of permitted research activities. Then we have confidentiality, protection of proprietary data and research results. And finally, liability and safety, responsibilities for material handling and safety. Keep this point in mind because we'll use it again later when we discuss research collaboration agreements, since many of these benefits overlap. This brings us to our next section, research collaboration and license agreements. This section covers two more important legal instruments that build directly on what we've already discussed, research collaboration agreements and license agreements. Let's begin with Research Collaboration Agreement or RCA. What is an RCA? A research collaboration agreement is a formal legal contract between two or more organizations that work together on a common research project. It clearly defines the roles, responsibilities, and rights of each collaborating partner. The key points here are: defines shared roles and responsibilities, enables access to shared labs and equipment, protects IP ownership for all partners, accelerates innovation and scientific discovery. Another important aspect is essential clauses in an RCA. The first clause is scope of work, defines the research objectives and the work to be carried out. The next clause is roles and tasks, specifies what each partner is responsible for. Following that, budget and funding, explains how the project will be funded and how expenses will be shared. Another clause is IP ownership, states who owns existing and newly created intellectual property. Then we have publication rights, defines how and when research results can be published. And finally, confidentiality, protects confidential information and prevents unauthorized disclosure. This is one of the most important concepts in this chapter because these 6 clauses are commonly asked as a list in examinations. Let's continue with benefits of research collaboration. The first benefit is shared expertise, access to diverse scientific knowledge and specialized technical skills. The next is better facilities, utilization of advanced research infrastructure and high-end equipment. Another benefit is funding opportunities, increased eligibility for large-scale research grants and institutional funding. And finally, faster research. Synergy between partners significantly reduces the time to achieve results. Before we move ahead, let's look at the challenges in collaboration. The first challenge is ownership conflicts, disputes over ownership of research IP. The next challenge is publication disputes, disagreements on authorship and timing. Following that, confidentiality risks, risk of leaking sensitive research data. Another challenge is resource sharing, challenges managing shared lab equipment. And finally, project delays, coordination issues causing timeline shifts. Notice how these challenges directly mirror the essential clauses we discussed earlier, which is exactly why those clauses are drafted in the first place to prevent these very problems.
One more important concept is the license agreement. What is a license agreement? A legal contract where the IP owner grants specific usage rights to another party under defined terms and conditions. The key features are binding legal terms and conditions, permission to use specific IP assets, grants commercial and selling rights, defines royalties and payment schedule. Let's continue with key elements of a license agreement. The first element is parties, the legal entities involved, licensor and licensee. The next element is licensed IP, clear description of the patents or technology being licensed. Another element is territory, the specific geographic area where the license is valid. Following that, duration, the period of time for which the rights are granted. Then we have royalty, details of payments, rates and payment schedule. And finally, termination, conditions and procedures to end the agreement. Remember this because it is frequently asked in examinations. These 6 elements form the backbone of any license agreement, agricultural or otherwise. Let's move to types of license agreements. If you observe the illustration, there are 5 main types. The first type is patent license. Grants rights to use a patented invention or technology. It allows making, using, selling, or importing the invention, protects the patent owner's rights, and usually involves royalty payments. The next type is trademark license. Permission to use a brand name, logo, or trademark. It helps in brand promotion and recognition, maintains brand quality and value, and ensures proper use of the trademark. Another type is copyright license. Rights to use creative works, publications, or designs. It covers books, articles, music, images, videos, designs, allows copying, distributing, or adapting the work, and requires credit to the original creator. Following that, know-how license, access to technical expertise and proprietary knowledge. This includes skills, formulas, methods, processes, not always protected by patent and usually kept confidential and shared through training. And finally, software license, permission to use computer programs and digital tools. It allows installation and use of software, maybe single user or multi-user, and includes terms for updates and support. The key takeaway here is each license type protects different kinds of intellectual property and grants specific rights for its legal use. Let's shift our attention to agricultural applications. Licensing agreements help transfer technology from research to real-world use, improving productivity, quality, and sustainability in agriculture. The first application is improved seeds. Licensing of high-yielding, drought-resistant, and pest-resistant crop varieties, increasing crop productivity and farmer income. The next application is dairy tech. Advanced processing methods, safe and quality protocols, and value-added dairy products. Another application is food processing. Better preservation techniques, innovative packaging solutions, and nutritional enhancement of foods. Following that, farm machinery. Automated and modern farm equipment, precision farming tools, and innovative harvesting technology. And finally, biotechnology. Genetic engineering applications, molecular markers, and development of useful biological traits. The key takeaway here is agricultural license agreements ensure that innovations reach farmers and industries, supporting food security and sustainable development. Since many of you are studying dairy technology, pay special attention to the dairy tech point here. This is directly relevant to your specialization. Let's continue with case studies in agriculture. The first case study is ICAR Technologies. Successful commercialization of high-yielding varieties and farm machinery through the ICAR AgriNovate portal. The next case study is SAUs and industry. State agricultural universities partnering with private seed companies for hybrid technology licensing. Another case study is dairy innovations. NDRI's transfer of milk testing kits and processing technologies to dairy cooperatives and private processors. And finally, startup licensing. Empowering agtech startups through low-cost licensing of public research to drive grassroots innovation. These real-world examples show exactly how the theory we've covered today translates into practice in the Indian agricultural research ecosystem. Finally, let's discuss the summary of IP management. The first point is commercialization, bridging the gap between laboratory research and societal application. The next point is legal protection, ensuring long-term security and rights for inventors and institutions. Another point is collaboration, driving innovation through synergy and shared research resources. Following that, material sharing, facilitating the global exchange of vital research materials via MTAs. And finally, agricultural innovation, modernizing the agricultural sector through structured IP management frameworks. And that brings us to the end of today's lecture. Thank you.